Welcome back. In the last class, we looked at how receptors are very important for the body in transmitting signals. In today's lecture, we look at the various functions and types of receptors. So, there are three major families of receptors. The first family is called the ion channel receptor, and the second one is called the G protein coupled receptor, and the last one is called kinase-linked receptor. All three of these are extracellular receptors, and we shall also look at certain types of intracellular receptors. So, the first class is ion channels. Okay. So, as we looked at earlier, the membrane is made up of a bilayer. Okay. So, it has a phospholipid molecules in the bilayer, and so what happens is that this makes the membrane quite hydrophobic. Okay. So, now the cells has an aqueous environment inside it which has many ions. So, these ions cannot cross the hydrophobic barrier because of the repulsion between ions and the lipids. Okay. So, for example, the movement of sodium and potassium ions across the membrane is really crucial for the function of nerves. So, this looks like an intractable problem. However, the way this problem is solved is that you have proteins which are embedded in the lipid bilayer, and these proteins, and as we know, proteins have made up of amino acids, and amino acids have polar functional groups, and these polar functional groups can interact with ions and enable their transport. Okay. So, the arrow here indicates a typical protein which can transport ions through it. So, these channels through which ions are transported are called as ion channels. Okay. So, ions therefore can cross the fatty barrier of the cell by moving across these hydrophobic channels or tunnels. Now, ion channels are actually complexes made up of typically 5 protein subunits, and these 5 protein subunits traverse the membrane. Okay. The center of this complex is hollow, and it is lined typically with highly polar amino acids, so that it provides a nice area through which the ion can go. Okay. Now, because this is arranged in this particular way, the ions can easily travel from outside to inside or from inside to outside. But if the channels are always open, there is no control. So, in order for there to be control, there is an extra mechanism that operates. So, what the analogy that we can look at is our gates. So, when a building needs to be secure or when a campus needs to be secure, what we do is we surround it by a wall. Okay. But now, we cannot pass through this wall, and therefore, there is gates that are installed. So, once if the adequate permission is obtained, the gate opens and you can enter, and the gate closes. Right. So, this kind of analogy is what we would use to understand how these kinds of ion channels are regulated. So, once the external chemical messenger transmits the signal, this is exactly what happens in an ion channel receptor system. Okay. So, in this cartoon, we shall look at how the ion channel receptor system works. So, in here you initially have an ion channel wherein it is locked. Okay. So, this is the gate that needs to be opened, and here is the cell membrane, which is a highly lipid rich region. And now, once a neurotransmitter or a messenger comes in, there is a site for the binding of this messenger. Right. And so, we already looked at how this binding is an induced fit model, and once there is an induced fit that occurs, this results in a conformational change which permits the ions to go from outside to inside. Okay. So, these therefore, the lock gate can be controlled by the messenger. However, when the chemical messenger binds to the external binding site, okay, so this in turn causes the overall protein complex to change shape, opening up the lock gate. Okay. So, now in fact, the receptor protein is an integral part of the ion channel complex. So, as you can see here, the log gate is over here and the receptor binding site is over here, and this together is a protein complex. So, any conformational change that occurs in after binding is also reflected in the log gate. So, the operation of this ion channel explains why a small number of neurotransmitter molecules can have a tremendous effect in the target cell. So, once you have the log gate open, typically thousands of ions can traverse the barrier. Okay. Therefore, the binding of a neurotransmitter to an ion channel results in not just a rapid response, but also a amplified response. Okay. The time of response is typically in milliseconds. So, ion channels are therefore the preferred mode of signal transmission in synaptic nerve systems. Okay. Ion channels are specific for certain ions. For example, there could be channels for sodium ions, potassium ions or calcium ions, which are all cationic, or they could also be anionic ion channels for chloride ion. Okay. The ion selectivity will depend on the amino acids that are lining the ion channel. So, for example, if you have amino acids which have a positively charged, let us say, an amine residue, they would preferentially bind to chloride. Okay. And it is also very interesting that mutation, what is mutation? Mutation is basically replacement of one amino acid with another, is sometimes sufficient to completely change the selectivity of an ion channel. So, therefore, the primary structure of these ion channels are significantly important for their function. Now, let us look into a little bit of detail of these ion channels. These ion channels are actually glycoproteins. What we mean by glycoproteins are basically these are proteins which are conjugated to sugars. Okay. And they have 5 subunits, and not all these 5 subunits are identical. So, here you have examples of 2 different kinds of ion channels which both have 5 subunits, and one of them responds is a nicotinic cholinergic ion channel, whereas the other one responds to glycine. So, the area, and these are controlled by, so for example, the nicotinic cholinergic receptor has 2 alpha subunits, 1 beta subunit, 1 gamma subunit and 1 delta subunit, whereas the glycine receptor ion channel is made up of 3 alpha subunits and 2 beta subunits. Over here is actually the light blue colored circle indicates the ligand binding site. So, in this particular case, this is where glycine binds. Now, in this glycine binding, the receptor protein in the ion channel that is controlled by glycine is the alpha subunit as shown here. Okay. Now, let us look at the individual protein subunits in this complex. Although there are various types of these, they all fold up in a very similar manner, such that the protein chain traverses the cell membrane. So, when you have a cell membrane, these subunits actually go across and come back and so on. We will look at it in a little bit more detail later. Okay. So, these domains are called the each subunit has 4 transmembrane regions which are hydrophobic in nature, and one can reason out why these need to be hydrophobic, because they need to interact with lipids on the membrane, and they are labeled as TM1, TM2, TM3 and TM4. They also have a neurotransmitter binding region, so here which is located outside, and these are the extracellular loops and these are the intracellular loops. Okay. And there is also a lengthy N-terminal extracellular chain which contains the ligand binding site, which I have already described just now. All right. Now, each of these subunits are arranged such that the second transmembrane region of each unit faces the central pore of the ion channel. So, if you look at it, this Tm2 is actually the one that faces the ions. Okay. Now, let us understand how the gating mechanism works. So, when the receptor binds the ligand, so over here this is the messenger and this is the receptor binding region, what happens is that there is a conformational change. Okay. First there is an induced fit and then there is a conformational change, and this conformational change results in ions flowing into the cell. Okay. This process is called as gating, and the binding of the neurotransmitter eventually opens up this central pore, allowing the ions to flow. This conformational change is quite complex and we will not be discussing this in this lecture, but what happens is that there is a sort of change that occurs which helps in opening it up. So, you can imagine that this is the area where the ion will not be able to go through. When these open up like this, then what happens is that they assume an open conformation which allows the ions to flow. Okay. So, eventually the central pore is through which the ions flow has to be created by conformational changes. Now, there are two types of gated ion channels. The first one, which we have already described just now, is the ligand gated ion channel. So, where the opening and closing of the gate is controlled by ligands. The other type of ion channels are not controlled by ligands, but instead are sensitive to differences in potential that exist across a cell membrane, which is also known as the membrane potential. These ion channels are actually present in the axons of excitable cells such as neurons, and these are called voltage gated ion channels. And voltage gated ion channels are really crucial to the transmission of signal along individual neurons, and therefore, they are very important targets for painkillers and local anesthetics. So, to summarize, receptors controlling ion channels are an integral part of the ion channel, and binding of the messenger induces a change in shape which results in opening of the ion channel. And receptors controlling the ion channels are called ligand gated ion channels, and they contain 5 protein subunits, with the receptor binding site being present in one or more of these subunits. Now, binding of a neurotransmitter to an ion channel causes a conformational change in the protein such that the second transmembrane domain of each subunit rotates and opens the channel. This allows the ions to go down. The next class of receptors are called as the G protein coupled receptors. To give you some idea as to how important these are, about 30% of all drugs on the market act by binding to these receptors. In general, these are pretty slow and they are activated by hormones, and these include the muscarinic receptor, the adrenergic receptor and the opioid receptor. And these are the time scales of these receptors are typically measured in seconds. There are a large number of different G protein coupled receptors which interact with important neurotransmitters such as acetylcholine, dopamine, histamine, serotonin, glutamate and noradrenaline. Other G protein coupled receptors are activated by peptides or peptide hormones which we looked at earlier, such as enkephalins and endorphins. G protein coupled receptors are membrane bound proteins that are responsible for activating proteins known as G proteins. G proteins act as signal proteins and they are capable of activating or deactivating membrane bound enzymes. Consequently, the activation of the receptor by a chemical messenger influences the reaction that takes place within the cell. So, let us look at this process now. Now, what happens initially is that the receptor is embedded within the membrane and it has a binding site. Okay. Now, on the inner surface there is another binding site which is normally closed, right. Once the messenger binds to the receptor, what happens is that there is an induced fit which helps the binding of the messenger. Once this binding happens, the inner binding site which was previously closed is now open. After it opens, it becomes a site for recognition by G proteins. Okay. And once the G protein, which is attached to the inner surface of the cell membrane and is made up of 3 protein subunits, but once it binds to the receptor, the complex is destabilized and fragments into a monomer and a dimer. Okay. Then these interact with membrane bound enzymes and continue the signal transduction process. There are several different G proteins which are recognized by different types of receptors. Some of the activated subunits from these G proteins have an inhibitory effect on membrane bound enzyme, while others have a stimulatory effect. However, the mechanism by which G protein is activated by fragmentation remains same irrespective of whether it is a stimulatory or an inhibitory effect. So, how does the signal get amplified? The amplification of the signal in this process, as one activated receptor activates several G proteins. Let us now look at the structure of these G proteins. The G protein receptors fold up within the cell membrane such that the protein chain winds back and forth through the cell membrane 7 times, as shown here. Okay. So, again, similar to the ion channel receptors, there are extracellular domains and intracellular domains. There is a binding site for the neurotransmitter or the hormone, which is on the extracellular portion of the protein, and the exact position of the binding site varies from receptor to receptor. There is also, as you can see, a G protein binding region which is shown here. We shall look into detail about both ion channel receptor as well as G protein receptor later in the course. The third type of receptors are kinase-linked receptors. Kinase-linked receptors are a superfamily of receptors which activate enzymes again, but they do not require a G protein. Tyrosine kinase are important examples of kinase-linked receptors. Tyrosine, as you know, has a phenolic residue, okay. And they are very important targets in developing novel anti-cancer agent. The response from kinase linked receptors is typically in minutes. So, in these structures, the protein concerned plays a dual role of both a receptor as well as an enzyme. The receptor here is embedded in the cell membrane, and one portion of this is exposed to the inner surface and the other one is exposed to the outer surface. The outer surface contains the binding site for the chemical messenger, while the inner surface has an active site in the resting state. Again, very similar to the other receptor ligand interactions, once the messenger binds to the receptor, it causes the protein to undergo a change in shape, and once this change in shape happens, the active site which was previously closed is now opened up, and this is an enzyme active site, and therefore, it can catalyze a reaction A goes to B. And reaction here that is catalyzed is a phosphorylation reaction, where a tyrosine residue is on a protein substrate is phosphorylated. We looked at earlier the various classes of enzymes that carry out various functions, and the enzyme that catalyzes the phosphorylation reaction is known as a kinase. Okay. So, therefore, this protein is referred to as a tyrosine kinase receptor. Here, ATP is required as a cofactor to provide the necessary phosphate group, and the active site remains open as long as the messenger molecule is bound to the receptor. So, therefore, several phosphorylation reactions can occur, resulting in amplification of the signal. These kinase linked receptors are activated by a large number of polypeptide hormones, growth factors and cytokines. Loss of function of these receptors can lead to several developmental defects or hormone related problems, and also overexpression can result in malignant growth disorders. So, therefore, these constitute important targets for drug discovery. Let us look at the structure of these tyrosine kinase receptors. These contain a single extracellular region, which is the N-terminal region, which also contains the ligand binding region, and the binding site of this chemical messenger is a single hydrophobic region that traverses the membrane as an alpha helix of several turns, and the C-terminal chain is inside the cell membrane as shown here, and this has the catalytic binding region. The important point here is that an external chemical messenger has managed to convey its signal or message to the interior of the cell without the messenger getting into the cell or without it being altered. There are other classes of receptors known as, which I mentioned earlier, which are known as intracellular receptors. And these are receptors, or about a 50-member family in this group, or 50 members of this group, and they are very important in directly regulating gene transcription. So, to recap, gene transcription is nothing but a process wherein something goes and binds to a DNA inside the cell and initiates the transcription process. So, these are are sometimes called as nuclear hormone receptors or nuclear transcription factors. The response from these intracellular receptors is in hours to days. Okay. So, when we look at the structure of these chemical messengers for these receptors include steroid hormones, thyroid hormones and retinoids. In all these cases, the compound or the messenger has to cross the cell membrane, get inside and then find the domain where it can bind to these receptors and lead to activation. They all have typically very similar general structures. They all have a steroid or ligand binding region, and they have a DNA binding region. We will look into this in a little bit more detail shortly, and here is the N-terminus. Now, these are a single protein containing all these various components. The DNA binding region contains 9 cysteine residues, 8 of which are involved in binding 2 zinc ions. Okay. Zinc ions play a very crucial role in stabilizing and determining the conformation of the DNA binding region. So, this protein has to bind to specific stretches of DNA. As a result, the stretches of protein are sometimes referred to as zinc finger domains. The DNA binding region for each receptor can identify a specific nucleotide sequence in DNA. For example, the zinc finger domains for the estrogen receptor recognize the sequence shown here, where A, G, C and T are adenine, guanine, cytosine and thymine respectively. Okay. Now, the mechanism by which the intracellular receptor works is very similar to the extracellular receptors. The major difference being that the messenger has to cross the cell membrane. So, once it crosses the cell membrane, it can bind to the receptor, and once the receptor ligand complex is formed, this can dimerize. Okay. After it dimerizes, of course, an induced fit take place first, which causes the receptor to change shape, and this in turn leads to dimerization of the ligand receptor complex. The dimer then binds to a protein known as the co-activator protein, and this together can go and bind to a particular region of the cell's DNA. Depending on the complex involved, the binding of the complex to DNA either triggers or inhibits the start of transcription, and this will affect the let us say the synthesis of a protein, and that protein may have important function inside the cell.